For so many people, the dream of starting a family runs into this incredibly frustrating, often heartbreaking wall. But today, we are diving into some absolutely groundbreaking research that's basically playing genetic detective, cracking cases that honestly were once thought to be completely unsolvable. And it all kicks off with this number, 15%. Get this, that's the percentage of couples around the world who struggle with infertility. It is a far more common challenge than most of us think, and it affects both men and women pretty much equally. And for a huge chunk of these couples, the diagnosis they get is unexplained infertility. I mean, can you imagine going through round after round of tests just to be told, we don't know why? It's the ultimate dead end, and it leaves people feeling completely powerless. But here's where the whole story changes. Because what if those answers weren't actually missing? What if they were just hiding, tucked away, in plain sight, written in the 3 billion letters of our own genetic code? Thanks to some seriously powerful new sequencing tech, scientists can finally read that code and solve these mysteries. So how in the world do they actually do it? It's one thing to find a clue, right? It's another thing entirely to prove it's the right one. Let's take a look at the really methodical toolkit these genetic detectives are using. It's really a 4-step process. First, they find a group of patients who all share a very specific, very severe problem. Next, they sequence their DNA, looking for a common genetic variant. That's their suspect. Now, this next step is absolutely crucial. They have to confirm what that gene does. So they create animal models, usually mice, and they see if removing that same gene causes the same problem. Only after they prove it can they even begin to think about new ways to diagnose and hopefully treat it. And this process, it is already leading to some massive breakthroughs. So let's open up our first case file. This one is all about male infertility and a gene that goes by the name of SINK2. Okay, so this whole investigation started with a family in France. There were 2 brothers, and both were unable to have children, but for slightly different reasons. The first brother produced 0 sperm. None. The second brother produced a very small number. So the big question for the scientists was, what on earth could be causing such similar yet distinct issues in the very same family? Well, after sequencing the family's DNA, the researchers zeroed in on their prime suspect, a single mutation in that SPINK2 gene, which both brothers shared. This, of course, led to the next massive question: what does SPINK actually do? So here's a cool analogy. Inside every developing sperm cell, there's this really powerful enzyme called a crossin. Think of it like a demolition tool that's needed to break through an egg's outer layer. The SPINK2 protein's job is to act like the safety lock on that tool, making sure it doesn't go off too early. And this is exactly what happens when that process goes sideways. In a healthy cell, the SPINK2 safety lock is on. It's engaged. Acrosin stays totally under control, and the sperm develops perfectly. But in the patient cells, that safety lock is broken. It's just off. Acrosin is unleashed inside the developing sperm, it causes total chaos, and it literally leads the cell to self-destruct long before it can ever mature. And this brings us right back to those 2 brothers. The brother who had a total loss of SPINK2 function, he had no safety lock whatsoever, which meant no sperm. The other brother, he had a less severe mutation, so his safety lock was just faulty. Some sperm managed to develop, but very few, leading to that low sperm count. It's a perfect example of how these conditions aren't just on or off. They exist on a whole spectrum. All right, let's pivot to our second case file. This time we're looking at a really recurring and, until now, mysterious type of female infertility. So this investigation centered on a group of 33 women who all shared the same devastating experience: repeated failed rounds of IVF. It didn't matter what they did. Their eggs just would not mature properly after the hormone stimulation. They were basically stuck in an early stage, which makes fertilization totally impossible. So when researchers sequenced their DNA, they found something pretty remarkable. More than a quarter of them, 26%, all shared the exact same mutation in a gene called PATL2. For the scientists, this was a massive, flashing neon sign of a clue. And this chart just shows you how dramatic the effect really was. In a typical control group, about 85% of eggs will reach that mature stage ready to be fertilized. But for the patients with this PATL2 mutation, wow,
A staggering 80% of their eggs were stuck, completely blocked at the immature stage. So what is PATL2 doing? Well, think of it like this. An egg cell is jam-packed with stored genetic instructions. These are called maternal RNAs that it absolutely needs to mature and survive the first few days of development. PATL2 is like the master librarian, managing all those instructions, making sure they're read at exactly the right time and in exactly the right order.
So you can imagine, without that librarian, you get absolute chaos. The egg's internal structure is just a mess, which kicks off a whole domino effect of problems. The chromosomes don't line up right, fertilization fails, and even if by some miracle it happens, the embryos that result are super poor quality and just can't develop properly. Okay, so these two cases are way more than just cool science, right? They really represent a huge, fundamental shift in how we're starting to think about infertility. Let's zoom out and look at the bigger picture here. Before this kind of research, you had patients stuck with that unexplained infertility label. Now they can get a specific molecular diagnosis. They can go from the absolute frustration of failed treatments to finally understanding the why behind everything they've been going through. And this is what paves the way to move beyond a one-size-fits-all approach and toward truly personalized medicine. So when you boil it all down, what this really means is that genetic sequencing is turning these painful mysteries into actual diagnoses. And for patients, just having an answer can be life-changing. It can prevent them from going through more useless, expensive, and just emotionally draining treatments. And for science, it blows the doors wide open for developing tailored therapies that target the specific genetic root of the problem. It really makes you stop and think, doesn't it? As we continue to decode the absolute fundamental building blocks of life, we're not just solving medical puzzles. We are gaining the ability to address one of the most basic, profound human desires. And it leaves us with this huge question to ponder. How is unlocking our genetic code going to redefine the future of family?